In the 1950s, a group of ranchers in Idaho were managing their flocks when they noticed a strange pattern during the lambing season. Their pregnant ewes were giving birth to lambs with a single central eye. This singular deformity appeared suddenly, affecting multiple animals across the region. Baffled by these Cyclops sheep, the ranchers reached out to the U.S. Department of Agriculture, asking for scientists to investigate the cause. The initial hypothesis was environmental. Researchers suspected the pregnant ewes had grazed on toxic, birth defect-causing plants found in the local flora. This physical anomaly turned a standard agricultural concern into a biological mystery that remained unsolved for decades. Scientists initially struggled to replicate the effect. They collected local plants and fed them to lab rats, but the results were inconclusive. To find the answer, one researcher lived alongside the sheep for 3 summers, directly observing which plants the pregnant ewes were eating. After 10 years of observation, they identified the culprit: wild corn lilies. The plant contained a specific molecule with 6 connected rings, which they named cyclopamine. While ranchers were told to avoid the lilies, the science stalled. For 40 years, researchers knew cyclopamine caused the deformity, but they didn't know how. The next step forward came 4 decades later from a team of biologists led by Professor Philip Beachy. Beachy's lab was studying the hedgehog gene. It's a gene found in many species, from mice to humans, that provides essential instructions for early development. When the team performed genetic modifications to turn off the hedgehog gene in mice, the results were severe. The mice developed significant defects in their brains and a single central eye. Then, while looking through an old medical textbook, Beachy suddenly saw photos of the Idaho Cyclops sheep from the 1950s. The similarities suggested that the two phenomena were linked. The cyclopamine from the lilies was likely interrupting the same biological pathway Beachy was studying in the lab. Genes guide development by telling cells to release specific proteins, which then relay instructions to other parts of the body. In normal development, the hedgehog gene releases a protein that binds to and inhibits a second protein called patched. This allows a third protein, smoothened, to send out signals. These signals tell the developing brain to divide into 2 hemispheres and form 2 separate eyes. When a pregnant ewe ingests a corn lily, the cyclopamine molecule enters this system. Cyclopamine binds directly to smoothened and effectively locks it up. This prevents the protein from sending the signals necessary to separate the eyes and the brain. A microscopic blockage of a single protein was the entire cause of the physical deformity seen in the Idaho lambs. This discovery had implications beyond agriculture. Since cyclopamine was highly effective at shutting down the smoothened protein, it could potentially be used as a targeted medical treatment. Researchers noted that some human syndromes involve a smoothened protein that is constantly active, predisposing patients to certain types of skin cancer. Early tests showed that pure cyclopamine was difficult to work with and caused significant side effects in humans. However, scientists used the molecular structure of cyclopamine as a blueprint to develop safer, more effective synthetic alternatives. These efforts led to the development of 2 FDA-approved skin cancer medicines in 2012 and 2015. By investigating an agricultural anomaly, researchers transformed a bizarre mystery into a life-saving medical breakthrough.